system transduces or converts pressure changes into action potentials that are carried by the auditory nerve to the brain. All right, we've got a few messages coming in. Like I said, don't be afraid to get it wrong. Just do whatever you think. Better to get it wrong now than during the actual exam. You could watch these later on the QC Learning Commons YouTube. It'll be recorded and then posted on there. That's where the last review session was also posted. I'll give you guys like 20 more seconds for this question. Yeah, I'll provide the link to it at the end. All right, so some of you guys responded, and yep, the correct answer is A, hair cells. So hair cells are the primary auditory apparatus used for um, understanding audio or utilizing audio into stuff that we could actually remember or like listen to. So stereocilia, which are on top of hair cells, depending on their direction that they're facing, they can either be depolarized or hyperpolarized. So as you can see on the right, they're facing towards the right, so they're hyperpolarized, so there's no signals going through. But on the left, when the stereocilia faces a certain direction, cation channels open, and when the cation channels open, the hair cell becomes depolarized, causing an action potential to occur. So good job to those who said hair cells. Now, when an individual rod cell is stimulated with light, its membrane potential, what does it become? 30 more seconds. All right, so a lot of you guys said A, which is correct. So one thing to keep track of, at least for rod cells, when hits rod cells, it has nothing to do with action potentials. A lot of students make mistakes where they think, oh, action potential happens, which means the membrane potential becomes more positive. Actually, it has nothing to do with action potentials, and it's the opposite in that the membrane potential becomes more negative. So in the absence of light, sodium channels are open, and that creates a dark current. However, when light hits the cell, rhodopsin absorbs the light energy, which activates a G protein known as transducin. That then goes to activate PDE, which hydrolyzes cGMP, and then the sodium channel is closed. Because the sodium channel is closed and sodium is a cation, and sodium can't rush into the cell, obviously now it becomes more negative, and so the correct answer is A, the membrane potential becomes more negative in response to light. So good job. If you brush against the doorway as you enter a room, you will trigger which of the following? And you'll have a minute. All right, 10 more seconds. All right, so there was a bit of a mix of responses, but so a lot of you guys got the correct answer, which is B. So the opening of a pressure sensitive cation channel. This ultimately comes down to memorization on the types of channels. So as you can see here, there's lots of receptors for different types of stimuli. So the receptor that we're talking about, touch, is a mechanoreceptor. So that's sensitive to pressure, and that makes sense when you feel pressure on your skin. Mechanoreceptors get activated, and it's a pressure sensitive cation channel. Thermal receptors are for temperatures. They're also a cation channel, and they're responsible for temperature. Depending on warmth, they open or close their cation channels. Electroreceptors are commonly found in like certain types of marine animals, where an electric current opens a calcium channel. Chemoreceptors are, for example, taste and smell. So, if you go back, G protein, if the question asked about a taste or smell, it would be a G protein. And then, because G protein receptors get activated after a taste or smell molecule binds to a receptor. And then photoreceptors are with light, and photoreceptors are responsible for secondary messengers. So you don't need to theoretically, you don't need to remember like the exact like details of like how every stimuli occurs, but what you do need to know is like the important details. So for example, if they asked for what type of stimuli requires a secondary messenger, the answer would be light. Or what type of stimuli requires a G protein, the answer would be chemoreceptor. But a lot of it, the type of receptor name, it relates to what the actual function is. For example, electroreceptor, obviously some sort of electric stimuli, thermoreceptor temperature, mechanoreceptor pressure, so you feel something on your skin. Any questions or should I move on? You could just put a thumbs up or anything. The recording will be shared on YouTube, like by the end of today, most likely. All right, thumbs up. All right, the next question. Auditory information comes from hair cells in the ear that are concentrated in the blank. Okay, so we actually have a mix of answers for this one. So the correct answer is organ of Corti. So, the organ of Corti is located within the cochlea. So when it comes to the other answer choices, tympanic, basilar, semicircular canals, vestibular apparatus, those surround the organ of Corti, and a lot of them are located within the cochlea. But the organ of Corti itself, which is this pink thing as you see here, that's what actually connects to the hair cells, and then those hair cells eventually have the stereocilia. Um, that obviously, depending on the direction, depolarize the cell and then transduces the signal. So this is more like a memorization based question. So yeah, the correct answer is the organ of Corti. The other the other structures surround it and they are part of the hearing, but the organ of Corti itself actually contains the hair cells. So how does tropomyosin control muscle contraction? And by the way, these numbers are the numbers that correlate to the sample exam if you want to check it later on. And I'll give you guys a minute for this. All right, so this also had mixed responses, but a lot of you guys did get it correct. The correct answer is C. So changes in tropomyosin conformation exposes actin myosin binding. So this is essentially what's known as the sliding filament theory. So when calcium is released from the sarcoplasmic reticulum, it binds to troponin. Then troponin, it shifts the position of tropomyosin, so it changes the configuration of tropomyosin, and that manages to expose the myosin binding sites on the actin filament over here. Then the myosin head manages to bind to the actin, and then the release of a phosphate from ADP initiates what's known as a power stroke. So that power stroke is what causes the sliding of the filaments past one another. And then after that, ADP is released from the myosin head, and ATP binds to the myosin head, which causes it to release from actin. Obviously, you don't want to stay contracted forever. You want it to like obviously go back to a relaxed position. And then the hydrolysis of ATP returns the myosin head back to its original position. And then if calcium is returned back to the sarcoplasmic reticulum, the muscle manages to relax. But if calcium remains available, you continue contracting muscle. So the correct answer, once again, was C, because troponin changes the configuration of tropomyosin and it exposes the binding sites. So make sure you guys understand this. I think the next few questions are all related to this. So what is the role of the sarcoplasmic reticulum in muscle contraction? I'll give you guys 30 seconds for this. All right, so the people who responded, all of you guys got it correct. Good job. The correct answer is A. It stores calcium ions for release during contraction. Um, as we just went over for the oh yeah, as we just went over for the last one, calcium is released from the sarcoplasmic reticulum. And so uh that's what helps cause muscle contractions. I believe I skipped the question. Yeah. So wait, I'll go over this one. Once again, in the presence of calcium, which is released from the sarcoplasmic reticulum, troponin changes the configuration of tropomyosin, and that exposes the binding sites on actin. So all of this happens because calcium is released from the sarcoplasmic reticulum. And then I skipped question 44, which you guys should also get. I'll give you guys 30 seconds for this. So, good job to the people who responded. The correct answer is C. Arrays of filaments overlap and slide past arrays of another filament. It's in the name of the theory as well, the sliding filament theory. So, as long as you know the name, you'd be able to answer this question. Any questions so far from anyone, or are we good? All right, I'll take that as we're good. And this also relates to it. So a hiker is bitten by a venomous snake. The venom prevents the release of calcium from the sarcoplasmic reticulum. So because of that, which of the following symptoms might the patient exhibit? So this is you have to apply your knowledge now. So I'll give you guys a minute for this. Someone asked if there was an easy way to remember the cycle. Unfortunately, I don't know if a easy way to remember the cycle, but the good thing about one thing you'll notice about a lot of the human body systems, for these past few chapters, it's very step by step. It's a very step by step process. You have step one, step two, step three. So nothing will realistically like change it a lot or change it like crazy. So it'll be a bit easier to at least remember it. At least as long as you know step one, you could kind of imagine what step two, step three will be. And then obviously try to remember the images because the images could obviously help a lot. And then a lot of you guys got the correct answer. So yeah, the correct answer is C because obviously when the sarcoplasmic reticulum releases calcium, that's what actually causes contraction of the muscles. So if the calcium is not released, then the muscles will not be able to contract. However, if the question was the venom uh, causes an overactive release of calcium, the correct answer would probably be between A and B. It would probably so that's another way you know it's not A or B. It's because both are extremely similar to each other. So they're probably that those are probably not the answers. But if the question did ask for a overactive release of calcium from the sarcoplasmic reticulum, the correct answer would be one of A or B. All right. So now we're gonna move on to the circulatory system. So the movement of O2 and CO2 between the blood in the tissue capillaries and the cells and tissues depends most directly upon uh yeah, I'll give you guys a minute. All right, so the correct answer for this question is D, the fusion of oxygen and carbon dioxide down a partial pressure gradient. B also has to do with pressure. However, B talks about total atmospheric pressure, which isn't true because it's actually a partial pressure gradient between high pressure or low pressure. It's not related to the total atmospheric pressure. So what happens is that red blood cells, as they go through, they take oxygen from high pressure and then send oxygen to cells that have low partial pressure. So at high pressure, a lot of the oxygen it's kind of compact, so the red blood cells are able to take the oxygen from the cell, and then at areas with low pressure, obviously the oxygen is not as compact, it's less dense, so it's able to spread around more, so it needs more oxygen in those areas. So then the red blood cell transports oxygen from places with high partial pressure and then sends them to places with low partial pressure. So which of the following characterizes the lungs of mammals? All right, so there was actually a lot of mixed answers for this one. So the correct answer is B, tidal ventilation. So tidal ventilation, it's not something that people commonly say, but if you didn't know what tidal ventilation meant, here's what you could do as well to try to figure out the answer. Air sacs is commonly found in birds, not humans. So air sacs would be gone. For C, complete emptying and exhalation, um, we don't completely empty our oxygen during exhalation. As you can see here, we have around a liter left of air within our lungs after maximum exhalation. And so tidal ventilation or tidal volume is just refers to the amount of air you exchange in breathing when at rest or while moving, etc. And then cross current airflow, that's commonly seen in birds, and counter current airflow, that's seen in fish, a lot of fish or reptiles, etc. So that's how you could kind of figure it out if you didn't know what tidal ventilation meant. But tidal ventilation just means the amount of air you exchange at rest. And that's commonly seen in the lungs of mammals. So humans often have difficulty breathing at high elevations, primarily because the blank than it is at low elevations. And I'll give you guys 30 seconds for this. All right, so a lot of you guys put the correct answer. The correct answer is D, the partial pressure of oxygen is lower. So similar to the way oxygen is transported in our body based on the partial pressure gradient, it that's also how it works outside in the regular environment as well. So A common mistake some people do is they put A, which is the percentage of oxygen in the air is lower. That's not necessarily true. In Within air, the composition of air remains the same, whether at high elevations or low elevations, it remains relatively the same. So the percentage of oxygen in the air remains the same. What the problem actually is, though, at the top of the atmosphere, because there's less pressure, air becomes less dense, which means it becomes a lot more spread out. It becomes thinner in a sense. However, at low elevation, sea level, etc., the air becomes more dense. So obviously, it's more compact. So when you breathe in, you technically breathe in more oxygen. However, like I said, at top of the atmosphere, the air becomes less dense because of the low pressure, and it spreads out. So the percentage of oxygen in the air, whether at the top of the atmosphere or at sea level, still remains the same. It's just how dense the air becomes. At the top, because of the partial pressure is lower, it becomes less dense and it spreads out more. And it becomes thinner, which is why when you breathe in, you're taking in less oxygen. Does anyone have any questions or all right. Which event can occur when fetal and adult types of hemoglobin are at the same pressure of oxygen? And I'll give you guys a minute for this. All right, so this is a hard question because I'm sure it's something like maybe the professor glanced over and it's dependent on like a specific graph which you may not have seen as well. So the correct answer for this is adult hemoglobin releases oxygen and fetal hemoglobin picks up oxygen. So I'll go over the graph first and then I'll also talk about how if you don't know the graph, how you'd be able to answer this question. So in this graph, as you see here, pressure of oxygen is at the bottom, oxygen binding is at the top. When we look at 20 PO2, as you see here for adult hemoglobin, at a pH of 7.2, it's around like 25% oxygen binding. For a pH of 7.4, it's around 40% binding. However, for fetal hemoglobin, it's around over 60% binding. So pretty much at almost all pressures, fetal hemoglobin requires more oxygen binding. So let's say you didn't know about this graph at all, how you'd be able to answer it. So obviously, for fetal hemoglobin, it refers to fetuses, babies, etc. So at that point, they need more oxygen because at that point it's a period of rapid growth and rapid development. So in order to support that rapid growth and rapid development, uh, fetal hemoglobin needs to bind to more oxygen and spread it to more parts of the body. So at the at the same PO2, the hemoglobin will have to pick up oxygen because it needs to rapidly grow, it needs to rapidly develop. And A is the only one that says that fetal hemoglobin will be able to take in the O2. The rest of the options says that fetal hemoglobin will either release it or E is too specific. So the correct answer is A. So now we're going to talk about which of the following animals would be the best for illustrating the evolutionary transition of the vertebrate circulatory system from a single loop system to one with separate pulmonary and systemic circuits. So I'll give you guys 30 seconds for this. All right, so we actually had a mixture of responses. So the correct answer for this question is actually C, lungfish. So the reason why it's not amphibians or reptiles is because amphibians or reptiles, they still contain both types of systems, whether as a tadpole or when they uh grow up to be either a frog, etc. So they still maintain both types. A lungfish, on the other hand, as you see here, lungfish have developed to have a partially divided atrium. So compared to a fish, a regular fish, a fish is a single circuit. So it has a heart with four chambers, and then the oxygenated and deoxygenated blood goes in one direction, just as in a simple circuit. However, lungfish have developed to have a partially divided atrium as well as a lung. And so the left side receives oxygenated blood from the body, and then it goes through the gill bypasses and it goes through the capillaries. And then the deoxygenated blood is what the right side receives. It goes through the lung, becomes oxygenated, then gets spread out through the body. So the lungfish actually showcases both types. The lungfish actually developed and evolved to show both types of circuitry for uh circulation of oxygen. So the correct answer is lungfish. Um, were there any questions or? See, I know amphibians and reptiles, like you would think it would be the answer, but that's because they still showcase both types, just at different stages of development. However, lungfish have evolved to show a double circuit compared to the original when they were just a fish. All right, move on to the next question. So, in the cardiac cycle of humans, blood pressure is at its maximum when, etc. I'll give you guys a minute for this. – 20 more seconds. All right, so the correct answer is C, ventricles contracting systole. So a lot of you guys, good job, because a lot of you guys did say C, and then some of you guys even mentioned like you were confused between C and A. So at least you know that blood pressure, maximum, contraction, and systole. And then relaxing and distal would be at its minimum. So it's good that you know that. The reason why it's not atria is because atria is just what receives the blood. So the atria in the heart is just what, yeah. So the difference between atria and ventricle, the atria is what receives the blood, the ventricle is what actually pumps out the blood. So the atria receives the blood from the body, the ventricle pumps out the blood from the body. So when the ventricle contracts, that's what causes systolic pressure, or when the ventricle relaxes, that's what causes diastolic pressure. So as you can see here, blood pressure changes are measured with I'm not going to pronounce that, and a stethoscope. So the systolic value, the pressure needed to compress an artery so blood cannot flow, so that's at its peak. And diastolic value, when it allows flow through the artery, and then that's resting. And this is all because of the ventricle, because the ventricle is the one that actually pushes. Um, the ventricle is the one that actually pushes it out, and the atria is the one that receives it. All right. So the pacemaker function in the mammalian heart is performed by the. So obviously this is a memorization based question. and I'll give you guys a minute for this. 20 more seconds. All right, so the majority of you guys got it right. The correct answer is A, the senoatrial node or the SA node. So I'll go over how this actually works. So an action potential is generated in the SA node, which is commonly known as the pacemaker. So then it spreads through gap junctions in the atria, which, like I said, receives the blood, and then they contract together. Then the action potential stimulates the AV node or the atrioventricular node, which then allows it to send the action potential to the ventricles. So it's contained within the atria, but then the AV node gets stimulated, which allows it to send it to the ventricles through both the bundles of His as well as the Purkinje fibers. So the Purkinje fibers bundle of His, their purpose is to spread the action potential through the ventricles. Um, the AV node, its purpose is to spread the action potential from the atrium to the ventricles. But the SA node, which is the pacemaker, that's where the action potential is actually generated for heart muscle contraction. So good job. In vertebrates, exchanges of substances between the blood and the interstitial fluid occurs in the. I'll give you guys 30 seconds for this. All right, so all of you guys got it right. The correct answer is capillaries. So this is like a definition-based question. And so in the capillaries, that's where fluid is either squeezed out or pulled back in and allows for exchange between the blood and interstitial fluid. So I'm going to talk about Starling's forces. It may be a question on the exam about what Starling's forces are. So Starling's forces are opposing forces that help maintain water balance in the capillaries. So there's blood pressure, which is when fluid is squeezed out of the capillary, so it forces water and small solutes out. And then there's osmotic pressure, which pulls water back into the capillaries. But these two pressures working in conjunction is what causes the exchange of material between water, solutes, blood, interstitial fluid, etc. So that's caused by Starling's forces. It helps maintain water balance in the capillaries, and it helps with the exchange of um, oh yeah, the review session will be posted, and it helps with the exchange of materials. All right, during periods of starvation, stored fuels are used in which order? I'll give you guys a minute for this. All right, so we had a mixture of answers. The correct answer is E, glycogen, fats, and proteins. So glycogen is obviously your sugar slash carb reserves. So those are what are used up immediately because that's what gives you the most amount of energy like quickly. So all your sugar, all your carb reserves gets used up the most. Then comes fats, which gives you the next most energy, and then lastly comes protein. So if we look at this graph here, so as you see here, carbohydrate reserves, they're depleted within a day without any food intake. So within a day, it's already gone. And then fat intake is next. And then as you see, protein intake does go down as well over time. However, fat intake drops rapidly compared to protein intake. And so on average, a person can survive 4 or 5 weeks without food. And that's due to their fat and protein reserves. Because at one point they have no more body fat left. And so they're not going to be able to survive. And then protein is then used at a serious rate with serious consequences. And you need protein, obviously, to support your body. So for people, if anybody, for like major bodybuilders, when they go into like a calorie deficit, um, would a person with more fat survive longer than? Theoretically, yes, a person with like a higher amount of fat probably could survive longer without food because they have so much more of a reserve to take the energy from. However, it still wouldn't be beneficial for them. And so, you're welcome. And so people that are in like a calorie deficit, for example, the reason people say, oh, if you're in a calorie deficit, make sure you eat a lot of protein, it's because when someone is in a calorie deficit, as you can see here, they lose a lot of fat, right? But if you're in a calorie deficit and you don't eat a lot of protein, you lose eventually over time, you lose your muscle as well. So any sort of significant gains that you've made in the gym, etc., they also get lost as well. So that's why when you're in a calorie deficit, people say eat a lot of protein. So you're still gonna lose some like muscle mass, etc. But if you eat a lot of protein, you'll be able to somewhat negate that and then make sure you only lose the fat. So movement of food from the stomach into the esophagus is normally prevented by blank. All right, so everyone got it right. Uh, good job. The correct answer is a sphincter. And so, um, there's two types of sphincter here, a sphincter and then the pyloric sphincter, and then I'll go over that in the next slide. So the sphincter is what actually blocks food from going down into the stomach. The pyloric sphincter is what prevents food from the stomach going into the small intestine. So they both prevent food from passing through. However, when it comes from the esophagus to the stomach, that is just a regular sphincter. And the pyloric sphincter is for the small intestine. So how the process works, after you chew food, the tongue, so the bolus just means the chewed up food. That's just what it refers to. So the tongue pushes the bolus to the back of the mouth, and then your sensory nerves initiate the reflex that allows you to swallow it. The soft palate gets pulled up, and the vocal cords are pulled together to close the larynx to make sure that food doesn't go in the wrong area. So the larynx is pulled up, covered by the epiglottis, and then the sphincter relaxes, which causes the bolus of food to enter the esophagus. And then peristalsis helps push the food down, and then you have the lower. I'm not gonna pronounce that. The lower sphincter, which helps prevent food from going into the stomach, and then eventually it goes into the stomach, and then the pyloric sphincter helps control the amount of food that goes into the small intestine. So, which feature characterizes insulin? I'll give you guys 30 more seconds. So good job. Pretty much all of you guys, yeah, everyone got it right. The correct answer is D, released in responses to increases in blood glucose levels. So, um, as you hear, when your blood glucose arises above normal, your pancreas is stimulated to secrete insulin. So that increases the insulin circulating in your blood, and then it causes an uptake of glucose by the cells, and then that obviously results in metabolic energy production, fat synthesis, and glycogen synthesis. However, when blood glucose drops below normal, um, when blood glucose drops below normal, the pancreas is stimulated to secrete glucagon. This increases glucagon within your body, and then that glucagon breaks down glycogen in the liver, which releases glucose to the blood. So if the question asked which feature characterizes glucagon, the correct answer would be released in response to decreases in blood glucose levels. So, someone asked if the correct answer would be B. It's not B because it would be released by endocrine cells of the pancreas, not exocrine. So it's like sometimes there are these small things that like trip you up. So D would be the correct answer, or it would be the best answer. So yeah, when you have a lot of blood glucose levels, insulin is stimulated, which helps uptake glucose. And then if you have low blood glucose levels, uh, the pancreas is stimulated to secrete glucagon. Glucagon breaks down glycogen in the liver, which causes more glucose to be released to your blood. And so those two work in conjunction to make sure your blood glucose levels remain regulated. Of the molecules listed, which is the most toxic to cells? And I'll give you guys 30 seconds for this. So the majority of you guys got it right. The correct answer is C, ammonia. So water is obviously not toxic. Sodium chloride, that's table salt, that's not toxic. And that's between ammonia, urea, and uric acid. So, uh, ammonia is too toxic to be stored. So nitrogenous wastes, which are NH2, they need to be released somehow. So most land animals dispose of nitrogen through either urea or uric acid. So humans, we release it through urea. Birds, reptiles release it through uric acid. And then urea can be stored in concentrated form, but requires water for disposal. That's obviously why when we drink water, a lot of our urine is urea and water. And then uric acid allows for water conservation. However, when it comes to ammonia, ammonia is too toxic for our bodies to store it. Only certain aquatic animals and fishes are able to store ammonia, but we're not able to do it, so we have to release it through urea/uric acid. So the correct answer is ammonia. Ammonia is very toxic. No, ammonia is not in urine. Or even if it is in urine, it'd be in extremely small amounts. The main foundations or constituents of urine is urea and a water. Actually, ammonia probably is in urine, but it's in extremely small amounts. It's not in anything like a large amount. It's definitely not the major amount that um is seen in urine. And so ammonia is the most toxic to land animals. All right, organisms living in a freshwater environment normally blank. All right, so we actually have a mixture of answers. So the correct answer for this is A. It excretes copious amounts of dilute urine and retains salt. So the reason for that is in freshwater fish, they gain water and some ions through food. There's an uptake of salt ions through gills. However, in freshwater, as you mentioned, there's a lot more water content compared to salt content. So there's more salt within the actual fish itself compared to the outside environment. Because of that, through osmosis, water goes through the gills and other parts of the body surface into the fish. And so, when the fish urinates, it excretes some small salt ions, but it excretes mainly water. And so, that's why it has dilute urine. Once again, because for freshwater, there's more salts within the fish than outside the fish. So water through osmosis goes into the fish. So as a result, when the fish releases urine, it's a large amount of dilute urine because it wants to retain the salts within its body. On the other hand, for marine fish or in saltwater environments, it gains water and salt ions from drinking seawater and through food. There's an excretion of salt ions from gills, and because the outside environment is extremely salty, osmosis is goes the other direction. Instead of going into the fish, it goes out of the fish through other parts of the body and through gills. And as a result, when the fish urinates, it excretes a large amount of concentrated urine, but a small amount of water. So, uh, seawater fish, they excrete really concentrated urine, while freshwater fish, they excrete really dilute urine because they want to for freshwater fish, they want to maintain the salts in their body. For marine fish, they want to excrete the salts in their body. So the correct answer was A. And I believe this is our last question. So, which of the following would you expect to find in a urine specimen collected from a healthy person? And this should be fairly simple. I'll give you guys 30 seconds. So yeah, everyone got it right. The correct answer is A, water. So plasma proteins and amino acids, if you see those in your urine, or if that's present in your urine, that could be a sign of kidney damage. Red blood cells, you obviously don't want to see blood in your urine. Glucose means that you have a high amount of glucose within your body, which could be signs of diabetes. And then water is obviously what you would expect to find from a healthy person. And I believe, yeah, that's the end of today's presentation. Before you guys leave, quickly, somebody asked if there's a chance that the practice questions will show up on the exam. On average, people usually see around like 3 to 4. It wouldn't be much, it'd be around 3 to 4. And so like I said, I had the numbers of the questions on my slides. Uh, if you go into the actual sample exam itself, there are lots of questions I skipped. The reason I skipped those is because most of them are mainly like memorization or memorization based or structure focused, and then the function of that structure. So I wanted to do more questions that required some application. Um, so yeah, the chance that these practice questions will show up, there'll maybe be around like two to three, but it wouldn't be a lot. And please fill out the feedback form. Obviously, if you guys were here for the last review session and now, I'd really appreciate it. That way we know what to improve on, what we could do better, or what went well as well. Where you could find the practice questions. So I believe Dr. Garofalo sent the sample exam on Brightspace along with the answer key. And if you guys do check the sample exam, the first 30 or so questions you'll probably notice are from your last exam. So you could skip around like the first 30 or so questions, but then everything afterwards is related to your final. You're welcome. So yeah, please fill out the feedback form. Helps us know what to improve on, what went well, what you guys think. You guys are very welcome. If you guys have any questions about the final, lab, etc., there's no sample question for exam four, so check exam three. Yeah, so only exam three. Yeah, so exam three is the sample exam that has, I think from maybe around like 35 or 40 upwards is 40 to 80 is around what you'll find for what you'll see in your actual final exam. You guys are welcome. I'm gonna stay on here. If you guys have any questions, feel free to ask, direct message, etc. And good luck for all of you guys on Thursday.